So from today plant pathology lecture will begin, and in this lecture I will discuss about the viroid. There will be many part, viroid part 1, part 2, part 3. So in the part 1 I will discuss about the some important feature about the viroid and the historical background. So talking about the some important feature about the viroid, they are the smallest causal organism, smallest infectious organism. And they has the smallest genome, in all the living beings. It has single stranded RNA, and this single stranded RNA is CCC. It means covalently closed circular RNA. And the main feature of this single stranded RNA is that it is non-coding in nature. Non-coding means they cannot synthesize any protein. So how they, if they don't synthesize any protein, so how they can harm the host? That will come in part 3 or 4. The molecular weight of this single stranded RNA is 1 lakh to 1.4 lakh Dalton. And we all know that viroid has no protein coat. Another important feature of the viroid is that some of the viroid actually show the property of ribozyme. Ribozyme is the RNA having the enzymatic property. That's why they do self-cleaving and ligation of genome from a larger replication intermediate. Because of this, it is proved that the viroid are actually escaped intron. You know, DNA has the exon and intron. So due to the property of ribozyme, this intron can come out and undergo some process from which the viroid actually originates. In later part, we will talk about the detail about the viroid origin. So now we will talk about the historical background. In early 1960s, the Raymer and O'Brien actually did the bioassay for the causal organism of potato spindle tuber viroid. पोटैटो स्पिंडल ट्यूबर डिजीज जिससे होता था उसका बायोएसे किया था. इन 1965 द रेमर एंड डाइनर ये प्रूव किया था कि द कॉजल ऑर्गेनिज्म ऑफ द पोटैटो स्पिंडल ट्यूबर इज नॉट टिपिकल विरोन. It is not typical virion. In 1968 the chrysanthemum stunt disease, chrysanthemum stunt and citrus exocortis are not due to some virus. This is proved in 1968. So later in 1971, Diner actually told that the viroid are actually the free single stranded RNA which has having the property of non-coding, and that's why he also named it as viroid. So in 1973, the electron microscope proved the hair pin-like structure of viroid. In 1974, from electron microscope, it is proved that the viroid DNA is actually non-coding in nature. In 1976, it is proved that the viroid has the CCC RNA, covalently closed circular RNA. And in 1978, first viroid was sequenced, and the viroid first which is sequenced is potato spindle tuber viroid. That's for today. In the later part, we will talk about the nomenclature and the structure of viroid.